Students, we shall discuss about some specific type of mutants in this class. So these mutants or these types of mutants are very important while we study molecular biology, genetics and all. So the first one we should know about is the auxotrophic mutants. What are they? These mutants have mutations in genes whose products are required to make an essential growth substance. They, therefore, cannot multiply unless that substance is provided as a growth supplement. What does that mean? Suppose E. coli. You have an E. coli strain which cannot utilize or which has a mutation in a gene that used to produce histidine amino acid, for example. So if you don't provide this histidine amino acid in your culture media, this particular strain will not going to grow in that media, or it will not survive because it has a mutation, and for that it cannot synthesize its essential amino acid that is essential for its growth. Here, for example, histidine. So to grow it, you have to provide or you have to supplement histidine into your culture media. So these type of metabolic mutants are called auxotrophic mutants. The original strain from which the mutant was derived can make the substance, can make the histidine, and can grow without histidine supplement. So this is called prototroph. Prototroph. Okay. Prototroph is what? This is a wild type. This is a wild type which has the ability to synthesize that particular metabolite or something, and it can grow well without supplementing that particular thing into its growth medium. While the mutant here, for example, this is his minus auxotrophic mutant. It cannot grow without you supplement histidine into its growth medium. So these catabolic mutants are very, very important when you study molecular biology. Other type of metabolic mutant or related to metabolism is catabolic mutant. These are actually the reverse of auxotrophs. This is the case where it's a mutant which cannot use a particular substance for growth that can be used by the wild type. That means, suppose the original bacterium here in this example can utilize the sugar maltose. Okay, and if you provide maltose in your growth medium, it can utilize it as a carbon source. But the catabolic mutant of this bacterium has some mutation in the maltose catabolizing gene, so that it cannot utilize maltose as its substrate. So this mol minus mutant is a type of catabolic mutant. So these are two types of metabolic mutants we should know, because we can use them for further study, for molecular biological study, for genetic study and all. So these are auxotrophic mutants and another is catabolic mutants. Other type of very important mutants that are used in molecular biology is conditional lethal mutants. What is lethal mutant? It's a mutation that actually kills the cell, right? So this is the lethal mutation. So think, if there is a lethal mutation, you cannot really isolate the mutant strain because the mutation will kill it. So in molecular biology and genetic study, we use conditional lethal mutants. What are those? These mutants are mutants, and these mutations occurred in some essential genes which are very essential for growth, but cells with mutations that inactivate the essential genes cannot be isolated unless the mutations inactivate the gene under only some conditions, right? Because lethal mutations will kill the cell, but that's why we use conditional lethal mutants. That means these are lethal but only under certain condition. So they are conditional lethal mutants. Most of the mutations, this type of mutation occurs in essential genes. Which are the essential genes? Those are required like for RNA polymerase, for required for translation like ribosomal proteins, DNA ligase, helicase. That means DNA metabolism proteins, right? So if there is a mutation in these genes, the cell will die, because if there is a defective RNA polymerase, the cell will not going to produce enough mRNA, enough tRNA, enough rRNA to survive. But so you cannot isolate them, really cannot isolate these lethal mutations. But if these mutations are such that they are lethal only under certain conditions, you can easily isolate them and grow them in your laboratory, and only under certain condition they are lethal, while in other condition they will going to survive, right? So these are conditional lethal mutations. These examples include some heat sensitive mutants, like there can be a mutation in the DNA polymerase gene so that it cannot survive, or it cannot replicate at 42 degrees centigrade, but it can replicate at 36 degrees centigrade. So think, you have a mutation in the DNA polymerase gene, and such a mutation inactivate this DNA polymerase, and this DNA polymerase cannot act as 42 degrees centigrade, but it can act at 36 degrees centigrade or 37 degrees centigrade. So this mutation is lethal when the temperature is 42 degrees centigrade, but not in 36 or 37 degrees centigrade. So this mutation is lethal when the temperature is 42 degree, but not lethal when temperature is lower, like 36, 37. So this is conditional lethal. So if you want to study how the mutations work, you can make a conditional mutant which you can grow at certain temperature, which is the permissive temperature. Here in this example it is 36 or 37 degrees centigrade, while it will not going to grow at its non-permissive temperature, that is 42 degrees. So these are conditional lethal mutants, and these are very, very important.